One. Hello!
Hi,
Welcome to Microbiology Twitter Journal Club, where we nerd out big about all things small.
my name is Danny, and in a previous life, I dropped out from my PhD in microbiology at the University of Chicago, where I was infecting skin I grew with MRSA. Nowadays, I'm a fact checker for pharmaceutical advertisers and the president of Biotech Without Borders, a nonprofit based in NYC dedicating to enabling the public with the tools of biotech.
My name is Faz, and I have a PhD in microbiology, and I've mostly worked on bacteria, but since then I've worked in academic publishing as a research integrity specialist, and now I'm currently working as an editor for a scientific journal.
Cool. Every week, we meet to talk about microbiology. Today is our news week, where we do an overview of papers that we've seen in the news, or other sources, to try to find an article that we want to cover next week in more detail.
And next week will be our deep dive week, where we take an article and we choose today, and we look at it figure by figure to learn what it can and cannot tell us. So make sure to subscribe to satisfy your microbiology curiosities.
And if you want to follow along with any of the papers that we discuss on either week, we link everything in our shared Zotero library in the doobly-doo below.
And we want to hear from you, so please use the comments or tweet us using the hashtag microdwjc hashtag, which is down there. Yeah, that's right.
Somewhere in that overlay.
Somewhere in that overlay, yeah. Um, but yeah, so I think we're going to start off with the vaccine news, because we've had some big vaccine news. The Russian vaccine has come
Yeah,
out.
the first data that we've seen from it, or the first phase 3 data at least. I wasn't aware that they were even doing a phase 3 trial. I guess I assumed that it was just getting trotted out into the population, but it's nice they do. I guess they did it simultaneously, right, with delivering it to their to people who wanted to take it.
Yeah, this is an interesting one because yeah, people are talking about it, and one of the things I quite liked about it were that it uses two vectors: the RAD26, which is a human adenovirus that's also being
Hmm.
used for the Johnson and Johnson vaccine that people are also getting kind of crazy about, and then it's followed up by
Hmm.
the adenovirus 5 vaccine. And
Yeah. So the theory behind that is maybe people remember that adenovirus is something that we could have pre-existing immune responses to. And even this idea of like when you get the first shot of the vaccine, right, you could develop an immune response to that vector. So like not even saying anything about the immune response coming from the spike protein, which we want the immune response to, but the way that we get the spike protein in. So this strategy is trying to make sure that I guess the second time you get the injection, you're not going to have an immune response to that.
yeah, and people are talking about mixed dosing for different vaccines, so basically giving you the primer of the BioNTech or and following up with a different vaccine depending on what's available. So
Hmm.
I think that this can be kind of instructive on that because it's also using two different vectors. But of course they're not quite the mRNA or the kind of
Yeah, yeah. I mean,
yeah.
I'm not sure if like it's always hard. Like, right? I think we've said this before. Like, comparing clinical trials is comparing like apples to oranges, right?
Mm.
Like, they're done in their own. They're controlled internally, but they're not really made to be. Sorry, I dropped my keyboard.
But yeah, they're not made to be compared. They're all designed to their own specific kind of guidelines that don't necessarily pass between different studies. I mean, the thing that is quite
Yeah.
interesting about this one is they do use the first dose as kind of they do a lot of work looking at how much a first dose actually does to prevent infection. So
Sure. Yeah.
so the idea of like single dosing. So like from the 21 days after the first dose of vaccine, they basically confirm that there's a difference between the vaccine and non-vaccinated group. So um,
Hmm.
seems really interesting. Um,
Yeah, and do they end up getting to like a final efficacy percentage in here? I'm seeing the graph, the Kaplan-Meier curve.
yeah. So I didn't get because they throw out a lot of different efficacy numbers through.
Yeah.
So like, they say they get 90% efficacy. I wasn't able to quite pick out the breakdown for that. Um, But I mean, in terms of actual like severe cases, they were a lot more like kind of clear that saying that there weren't any moderate or severe cases in the vaccine group, which I think,
Hmm.
yeah, which
Which
is
I mean, those are very rare too. Like even in the big studies that we've seen, right, with several thousand, um, there have only been one or two of the severe cases, which is always good.
yeah. So I mean, so it's interesting. I mean, it's good to take into account, but again, I think taking your point that they have been giving this vaccine out to lots of people by now, we would hope to see something akin to phase 4 at this point because they have went ahead with it
Yeah. Oh,
and
I see. I see. table 2, they have the interim results on vaccine efficacy. That gives us a bunch of different things. Right, but most of it's from the first dose, and they only have a little bit after. They only have the all vaccine efficacy from the second dose. It looks pretty good.
It looks pretty good. I mean, yeah, it seems good. I'm not sure, like, whether we can really gain anything from going too deep into it, but
Yeah,
because
I mean, it's out there.
it's out there. You can read this. We've got all the links to all the papers we'll be discussing today in the comments. So if you want to read it for yourself and pick that out, then you're free to.
Yeah, I mean, I was just thinking, like, it's also just interesting, right? Like, this is also something where it's being out there, right? Like,
Yeah.
the policymakers who decided on using this vaccine, like, almost didn't care, right? What this result was going to be, because I guess for their risk benefit analysis, right? It was like this had to get out. I'm sure there's other reasons. There's always other
Yeah.
reasons for
Yeah, I mean,
something like this to
there's lots
happen.
of different reasons, but I can sort of see what. I mean, it's one of those things where you can really claim a big victory in hindsight if it turns out this vaccine is really, really useful, because
Right,
then you can,
right.
if you can pair that up to all the data of like people who were vaccinated and be like, okay, well, we've seen this had a tangible effect on the population, then that's
Yeah,
it was a
yeah,
really big gamble, and I don't
right.
think that they were really right to make it, but I think that um, it, I
I,
mean,
I, I, I totally agree. And I just wanted to point out because like, the data looks so good and we're like, oh, it's all fine, but it's like this is in hindsight, right?
yeah, I mean, I've seen the film Last Man. Oh wait, so was it the
Oh, I Am Legend?
I Am Legend where a vaccine goes crazy and turns everyone to zombies, and we narrowly avoided that this time. So I'm, but
Okay,
anyway,
so
but
we
yeah,
have
this is
single
positive
dose
news.
single dose administration and the influence of timing. Uh,
Yes,
Booster dose and immunogenicity of the Chadox and COVID-19 vaccine.
yes, single dose. So this is interesting because this is a follow up essentially to the previous vaccine paper. It's got the same authors, and they follow up on the vaccine trials, and more data has been collected since then. So it includes data from all four trials this time. So
Yeah.
last time they were missing a bit, and they
Yeah.
also,
I think like a big part of the UK trial, I remember.
yeah,
They
uh,
had mostly Brazil group.
yeah, and they revisit the low dose standard dose group, which is the group that I think gave us a lot of
Yeah.
the group of like extending errors of this like that have
Right.
snowballed.
Yeah. Strange, fortuitous, although fortuitous, right? Like because
Yeah,
it showed potential promising result.
yeah, and so they found that this group still has quite a very positive result. Um,
Hmm.
so I think they do a little bit more analyses looking at the efficacy of the single dose,
Mhm.
and looking and I guess taking into account the data of so taking into account the accumulation of cases after single dose, because there was a long delay in between them, so you can do some data analysis to figure out
Yeah.
whether it was more protective or not. Um,
Mhm.
so I mean, at the moment, it's not the paper's not in its best form. I think it'd be good to look at it after it's been published formally.
Yeah, I'm
But
I'm scrolling through it a little bit right now. I mean, it looks kind of like it's very much this is the same people that published. This is the group that brought us Chadox, right?
yeah,
And they're giving us an update almost of their data, reanalysis and update.
yeah. So it's still in preprint,
Mhm.
and it'd be interesting to see how it looks in its final published form.
And they don't speak to. I don't see them speaking to the asymptomatic, right? Because they had done there was like at-home swabs for one of the cohorts.
Yeah, I mean, I think they do look into asymptomatics, but they don't find any differences in those groups, or
Hmm.
they don't find. So that was a bit of a puzzling data because it seemed like that what the graphs were saying didn't necessarily match what their data. But that's because I didn't get the time, because I think the graphs were quite complicated, so I didn't get the time to understand them properly. So I think that
Yeah,
was more. It was. I think that I needed to dig into data to figure out what they're saying about it. But I think
right. Oh yeah, I see. In this table here, they do have asymptomatic COVID-19 cases 14 days after the second dose. Cool. Okay.
yeah,
Yeah, I guess we won't know until we dig in, but it's nice to know this info is out there. I
yeah,
guess I'd be interested to see it after it gets all tidied up.
yeah.
Yeah.
And yeah, again, we've got more preprints. Again, focusing on the idea of the first dose and whether it can reduce infection. This
Mm.
time, this one's for the so the effectiveness of the first dose of BNT162b2 vaccine in reducing SARS-CoV-2 infection 13 to 24 days after immunization. Real world evidence.
Mm. That's a big buzzword in yeah, real world evidence. Because it's just like I mean, yeah, anyways, it just sounds better, right? Like of course you want real world evidence, but like these are done in studies, right? These are studies that are being
Yeah,
done. And the further they get from the clinical study framework, right, the more they become these prospective or like longitude, right, like kind of not as controlled ways, like open label. They move into that realm where you can't put the right placebo controls in. So, anyways, I just I just
yeah.
want to say that that word has a bit of a buzz ring to it, but I mean, it does describe something like in a population of people that have varied, pre-existing conditions and so forth.
Yeah, I mean, because it really depends. Because again, they in the clinical trials they do specifically look for people with some conditions for it, or look. I mean, real world evidence kind of implies that clinical trials they're not real people they're experimenting on, which is a bit disingenuous. So, but I mean, I think the idea is like this is after it's been rolled out. So essentially, this is like being pitched as a phase 4 study, a retrospective cohort study. They're looking at the
Mhm.
rollout of vaccines to the public and seeing how that affects the spread of COVID in
Yeah.
people who got the vaccine versus people who didn't. And what they find, they calculate based on like the incidence of PCR confirmed SARS-CoV-2 infections. There's like a 50% reduction, 13 to 14 days after the first dose. So they do this weird thing. I think a lot of these studies doing that when they're studying the first dose is that they basically they these vaccines take some time to get to work. So after you get
Yeah.
the vaccine, it's not instant protection. You have to wait a couple of days until it gets protection. You get that protection. So
Hmm.
in this case, they look at the BioNTech study and they said, okay, well, those first 12 days, the patients aren't protected. So let's divide our patient cohort into people who who caught COVID in the first like 14 days of the vaccine, and then the second one of people who caught it afterwards, and seeing whether
Mm.
those two numbers were similar or not. Um,
Compare, yeah, I see. Compare the two groups in that window, right? The Lou time, effect, like maybe the establishment of efficacy window, we'll say.
Yeah. So the idea is that after 14 days, efficacy has been established. So that group is fundamentally different from the other group. So you can use
Hmm.
them as almost controls for each other and say like,
Hmm.
okay, this group they've been vaccinated, but we're going to treat them like they're the unvaccinated group, and this group they're vaccinated as well, but this is where they got the immunity. And so they did find
Yeah.
a difference between those two groups. And so 51% doesn't sound great, but that is still quite a useful amount of protection.
Yeah, absolutely.
But I
So
mean,
I
this
guess it's like it's interesting. I was just trying to think like because I've heard a lot of this news right about like oh maybe you can make vaccine stocks go further by just giving people one,
yeah,
right? This is evidence along that line, trying to convince people, or if I guess if there's some need, right? I mean, maybe this goes back to our modeling when we were reading that modeling paper, right? Like there could be a need to do some sort of intervention in this way, and
mm,
and this would allow people to understand what that trade-off was of just giving people the one.
yeah. And I think this brings it. So this is a preprint, and the next paper is also a preprint. But the funny thing is that this caused some controversy, and these other researchers have beef with this one. They've got
Ah,
so another group of researchers published a study called "Estimating the Effectiveness of Pfizer COVID-19 BNT162b2 Vaccine After a Single Dose," a reanalysis of a study of real-world vaccination outcomes from Israel.
right.
So,
So is this the same data set then? This is the same data set they're
oh,
using?
I'm gonna get to that. This is theoretically the same data set, but I'm gonna get to that because this is
Okay.
really interesting because um, so again, this paper comments on the previous one. They try to reanalyze the same data, except they don't have access to the same data. They didn't have access. So what they did was they used Microsoft Publisher to measure the graphs and then extrapolate the data from the graphs.
I see.
So they didn't have access, but they did like break down. So these graphs, they have a Kaplan-Meier graph. So if I can, so I'll pull up the original study. It might take a while to open up, but um, But yeah, with
Right.
Kaplan-Meier graphs, it's a stepwise study. So every step you get is equivalent to patients like either dropping out of the study or dying.
Right.
Um, so theoretically, if you can have a measurement software, so that you can actually do this with other bits of software as well that are
Yeah.
designed
Anything.
to it. And I know I've used that to review papers in the past, so that I can quantify like so so
This
what.
is the difficult, like when people don't publish their data sets openly, this is like the hoops that people go through in order to extract the information and do some science on it.
Yeah. So this is so I'm not necessarily against this, but I do understand how that's kind of difficult.
Yeah.
So what they did was so in the previous study they broke this into two groups, like we mentioned. This study they treated it almost as if the immunity was building gradually. So so
Hmm.
almost say break it into small buckets and look say, okay, on this day, what's the probability of the person catching COVID? And on this day and this day and this day sequentially. And they graphed that to try and what they say is that actually breaking it into two buckets oversimplifies the data. So if you like say looking at a graph and you that's going steadily up and you cut it down, then the averages of each point will not necessarily reflect what the actual data looks like.
Yeah. Okay. Makes
So
sense.
the idea is that so what they say is that by the final day of vaccination, your protection is actually close to say 90% because
Hmm.
they've broken it into small buckets and saying okay on that day your protection is built up to this point.
Right.
So they
But those
almost
early days are not so good, and the high efficacy of the later days, right, pulls that whole average up into the middle.
yeah. So yeah, effectively they treat it as a continuum, whereas the other group treat it as two different pop boxes. And
Right.
and I think the second group might have a point that actually the building of immunity doesn't happen the same in everybody, and there is like a it does build up gradually. It doesn't necessarily like switch on and off. Um,
Yeah. Mhm.
but
Which I
this
mean
is
that's more biologically consistent, right? When you think about how the body might work, right? Like some people might be getting it earlier, but the majority of the people are developing it as you get closer to that point where we say after that point they have immunity.
yeah. So this is a really interesting kind of example of like why preprints are really interesting and kind of important because this is almost like a really detailed review that you'd probably never get if you're just reviewing for another journal. But since there's a preprint that was released, suddenly there's all this
Yeah,
commentary that's happening, and I'm hoping that in the finished paper, that all of this can be synthesized together to create something quite interesting.
right. I mean, I think this is also speaks to the difficulty in media coverage on this, right? Because
Yeah.
what you get is two headlines that say, right? You get two titles, right, that sort of contradict each other in a little way. And that makes it
Well,
confusing to dig in and say what's going on actually.
yeah, I do feel like they have been playing these two papers off. Even they, because like the paper that the first paper was very much like, oh well, there's still some protection. The other paper is like, no, there's more protection. You're underestimating it, and I'm like,
Yeah,
okay, I I can understand why you'd want to underestimate things because of precautionary principle. You want to be like kind of secure and be as pessimistic as possible. But on the other hand, I feel like the second one's analysis are more biologically relevant. But then again, they don't have access to the real data. They've had to come
yeah,
out with like a
yeah,
ruler to try and measure the points. So might not necessarily be as accurate as they hope. Um,
yeah.
so
My hope is that like there's collaborate, or right, or like this because this idea is out there, then the original group can pick up this idea, right, and work on it. I mean, that's
yeah,
like the truly collaborative nature of science, right, in that way.
I mean,
Um,
yeah, you'd hope that. You'd hope that they'd collaborate. And I mean, the nightmare scenario is if they try to incorporate these data points and the other researchers go, well, no, you can't. We published this in our preprint. It's ours now.
yeah, yeah.
And yeah, so I'm hoping that they're thinking about being like higher and like helping everybody rather than thinking about. But yeah, it's.
So,
But yeah,
all
it's
right,
something.
what's next on the docket?
Yeah. Single dose vaccination in healthcare workers previously infected with SARS-CoV-2. Now, this is something that's quite interesting. So, let's say if you've had COVID-19, should you get vaccinated and will it do any good? Um,
Hmm.
so it's a really short paper, but the basic premises of is going to people who have had COVID, testing for whether they've had it in the past, and seeing
Yeah,
how that affects level antibodies. And
interesting. Yeah.
And yeah, and I think what they kind of find is that it almost acts like a booster to that original infection. And so they've
Yeah, it
got,
does seem at least the antibody titers are much higher, right?
yeah.
As people get vaccinated.
And I guess one thing that might be interesting is if people get more side effects after their boost, if they've already had COVID 19. Because I think people have been reporting that, and I feel like that might be something that happens.
I
Uh,
see, like a potential, yeah, increased adverse events or something from that vaccine dose.
Yeah, but then I'm saying this because there's a personal bias. My parents got vaccinated, and we had a COVID scare earlier in the outbreak. So, and when they come back with symptoms, part of me thinking, wait, that's a lot more than I was expecting. Maybe that's because that was COVID we had previously. But
Sure.
but then again, that's just an anecdote. So we
Yeah,
don't. That's not data. But it's interesting just in the debate of like where should people who've had COVID be in the vaccination queue. Um,
sure.
yeah,
Yeah, no, very interesting. But again, like just neutralizing spike, right? Titers of neutralizing spike antibodies is, so yeah, I mean, I guess that's the best information we have now. And it's going to be some, longitudinal study that's done afterwards following this cohort maybe, right? And seeing their
yeah.
incidence. If they even have the resources to follow this cohort of people that they studied.
Yeah, I mean, I guess it'll be something that'll be done in retrospective analyses like several years down the line where we'll pick that
Mm,
up. But
yeah,
yeah,
yeah.
it's an interesting idea.
Well, I was just gonna say this is also like my personal story, right? Like my grandmother got COVID and then they gave her the vaccine right afterwards.
Mm,
So I don't know. They just throwing things at the wall, I guess. She's fine, by the way.
yeah. Okay, that's good to know. So yeah, moving
Okay.
on for that. So, do antibody positive healthcare workers have lower SARS-CoV-2 infection rates than antibody negative healthcare workers? So, we're still operating off of the antibodies, but now we're going back to the does it does having COVID protect you against reinfection? Um,
Yeah.
and they
I was
yeah
gonna say I feel like we covered something very similar to this, but this is like a very large study now being done
yeah
specifically for this purpose.
yeah. There's like 14,173 participants contributing
Mhm.
1,868 646 days. There's lots of days of follow up in this. There's a lot of
Yeah.
data here, and it's so yeah. This basically adds to that study that people who've had COVID tend to be more protective. They do get some reinfections, but they're still quite a low level. Um,
Yeah.
so
Oh yeah, because they can even look at the reinfections as they give the CT ranges for the reinfection. So you can see like a approximate viral load, I guess, is one of the thoughts.
yeah, I mean this is an interesting thing because it's so big, we actually can get like some solid data on those reinfections. Because early on in the outbreak, we knew that they existed, but they only came out in case studies, and now it's gone bigger
Yeah.
and bigger. They've
hmm
figured out a definition for how to define when someone's been reinfected or not.
hmm
So
So
there,
what is that probable positive cohort? I'm trying to understand. Do one of these percentages give us that reinfection percentage?
I
Just
can
just from the whole cohort. It wouldn't, and it's only 362. Oh, that's all of the reinfections. Oh, I see. These are all the reinfections. I'm looking at table
yeah.
2 right now. I see, and then they do a frequency of new infections. Yeah, I mean, this is interesting stuff. I mean, I've been so curious about this this question. It's nice to see like a large population and to look at this because I was also, yeah, all those case studies. You know what happens? You figure it's probably rare because it's case studies,
Mm.
but yeah, trotting out the diagnostic tests to answer this question is really interesting.
Yeah. Um, so this gets us start on the question of immune memory. So like after you've had COVID, like how does your immune system like remember it?
Mhm.
So that brings us to a science paper, sorry, I keep immunological memory to SARS-CoV-2 assessed for up to 8 months after infection. So so yeah, immunity is more than just antibody responses. There are like memory cells involved. There's so this one actually takes the time to try and it measures the decline of antibodies over the time, but it also looks at like memory B cells memory CD8 cells and memory CD4 T cells. So it does
Yep.
the three immunological compartments, and they look at like the I think they longitudinally look at subjects over like 5 months after infection to see how these compartments are
Yeah.
maintained.
I mean, this is like a, in some ways, is an expansion, right, of observations people were making of like just antibody titers,
Yeah,
right? But now they're also going to look in these other immune compartments, right? T cells and B cells. Memory too, they like tag them so that they're the memory cells that they're measuring.
yeah. So
Hmm.
it's
I can't relate. These graphs are so dense. I don't even get anything from just looking at them, from just browsing through them. I'm just like, my goodness. So there's like it does decrease, but I guess what's going to be important in this is like the rate of their decreasing, or maybe like the specific subsets that they're measuring. They
yeah,
maybe see bigger decreases in some versus others.
yeah. There's a bunch that yeah it's there's a bunch that we could dig into here.
Yeah, it's like a big inventory, right? It's a big inventory of immune the immune reaction as it goes out. But they don't have them re-challenged necessarily, right? This is
Mm.
actually, they probably don't even know what's going on. Maybe that's something we would find out if we dove into it, right? I'd be interested to know, like, did they do they have people in their cohort who like did report a reinfection or something? Maybe it's too rare of an event, right? But that would obviously change the profile of what they're looking at.
Yeah, I think that would be interesting. Because I don't think they had a big enough cohort for that.
Yeah, I don't think they do either. Otherwise, you would see some sort of like subgroup thing in these figures, but it doesn't seem like that. It's just like one big cohort, um, variable,
Yeah.
like all humans are, slowly decreasing in different metrics.
Yeah, so they do test with like spike protein, see how they react. Yeah, that's
Hmm.
interesting. Now the next thing we're going to go back to a topic we've been visiting on quite frequently, the B.1.1.7 strain. So this is a new strain that's come out in England for in like December, and
Hmm.
last time we had this news show, there was like quite a lot of panic because the UK government had went out and said, okay, we're worried that this is much more severe. There are maybe more deaths associated with it, and
Hmm.
they cited three papers that were as of yet unpublished and still are unpublished at this moment. But so in the meantime, to follow up on that story, we've got this study coming from the COVID Genomics UK group. They've been looking. They pulled data from this thing called the COVID Symptom Study app, which is essentially this app where people would enter in any symptoms they're feeling on a day-to-day basis,
Hmm.
and then and these people there are people around the country who around the UK who have this app, and then they're
Yeah,
comparing that to genomics data collected by COG around the same time. So
interesting. Yeah,
the idea
to
would
try to
be
correlate those
yeah.
symptoms that are appearing with the emergence of the new strain.
Yeah, so the idea that okay, if we let's take a look, and if this area has lots of new strain, are there any changes in the symptoms? They didn't really seem to find anything like that. They didn't find any change in this disease severity, and they also tried to look at the potential for reinfection as well. Didn't really find anything there. So, and it's
Fascinating.
interesting because they're pulling from the same Public Health England data that the previous study seemed to be pulling from. So it'd be so I'm quite curious now to see what those studies have found and
Sure.
see, but yeah, either way, regardless, I mean,
It's
it's
very far. It's kind of interesting, and like it's all this. This is like what we've been seeing with the new variant, right? Is that
mm,
like you have to get really creative with the way that you're collecting the data because like there's lots of people infected. We know there's a large proportion of people infected like this, but like we don't have those like bench research experiments, right, to
mm,
correlate things. But yeah, it's very clever, I think, to use that survey. But I also see that it could potentially introduce a lot of artifacts and just be a bit uncertain in general, like
yeah.
what's going from there.
I mean, if there's like a say a bad flu outbreak in one area, something that might change things, or it might.
Yeah,
So there's a lot of different confounders in there.
yeah,
But I found it kind of interesting.
yeah.
Next up, increased resistance of SARS-CoV-2 variants B.1.351 and B.1.1.7 to antibody neutralization. Um, so we saw
Mm,
some information last time around that was quite scary for the B.1.5, sorry, the B.1 B.1.351 strain, which is the South African strain that's been referred to, and also
Yes.
the N501V2 strain as well. So
Which is one of the mutations in various of the variants that have appeared.
yeah, so this has mutation in the spike protein, and this adds to the data with respect to antibody neutralization. They focus down the contribution of different spike mutations. So they look at some monoclonal antibodies generated in prior studies, so including stuff like the Lycov, which is an antibody monoclonal antibody treatment. They also look at like Regeneron, and
Huh.
they checked that against convalescent vaccine serum. So and they looked to see like what part of the spike proteins are being neutralized, and neutralization doesn't seem to get abolished completely, but the South African strain does seem to have does seem to tend to need more antibodies to neutralize it than anything else. So we are seeing that slight resistance. So which is
Yeah, which is confirming that sort of the frightening result from the convalescent serum, right? That
yeah,
we saw from the South African variant. Yeah. Oh, they're also looking at convalescent plasma in this one as well.
yeah.
Yeah.
So so this one has a
So they
bit of
have the
everything.
same. They also have a replication of that data that we saw earlier because they
Yeah,
have convalescent plasma over the South African strain.
yeah, and replication is very important because each study doing having more people investigating a research question, it does kind of have that add that confirmation.
Yeah.
Um,
But this convalescent is these aren't this is not convalescent meaning they've been previously infected, right?
yes,
These people have been previously infected. But there's also they also have vaccine. I've never said that word before.
yeah,
Vaccinee serum.
yes.
Um,
That's getting more important because now people are worried about whether vaccines can protect against these strains. Um,
right.
yeah, which gives me the perfect segue to the next paper,
Yes, absolutely.
which is SARS-CoV-2 B.1.1.7 escape from mRNA vaccine-elicited neutralizing antibodies.
Mm.
So
A very grim title.
yeah, so can B.1.1.7 resist vaccination? So they take data from single immunization, and they measure like antibody responses against the wild type and then against mutant. So I think they take blood sample from people who have just been immunized once, and then they look at to see whether there's any reduction in the ability of sera from vaccinees to inhibit either the wild type or the B.1.1.7. And
Mm.
they found that that yeah the vaccine sera seem to display some high neutralizing uh, antibody activity against the new strain, but then when they looked at monoclonal antibodies, they found something very different. So
Mm.
they tested like different monoclonal antibodies, and they found that they could be so the B.1.1.7 was able to escape neutralization by a lot of the N-terminal targeting the monoclonal antibodies.
I
Um,
see. Like a specific region is not so good to be targeted.
Yeah, but when you've got like a convalescent serum, which is a milieu of lots of different antibodies, that the effect doesn't seem to be there. So I found that quite interesting because I think a lot of this,
Yeah.
yeah,
That is an interesting phenomenon because like we know that there probably would be, right, a different antibody profile that comes out from vaccines versus convalescent serum.
yeah,
Um, but what is that? Is it? I'm trying to understand if that means that they think that. I mean, this would be a reason to get vaccinated, and even if you have been previously infected, right? This is also
yeah,
part of that sort of thought, because like it really is a very different profile from the antibody side. And again, like I think maybe the thing that we should remind people of is this is that one component of immunity, but there's also that cellular component that's just more difficult to measure and hasn't been pulled through in this
yeah.
type of analysis.
Yeah, definitely. I think the thing I wanted to put out is the idea that the main thing that whether these viruses these like antibodies failed was when they're using monoclonal. So monoclonal is just one antibody. There's lots of it,
Yep.
and it's focused on a specific point. So a mutation in that point can mess up that monoclonal antibody. Whereas the the antibodies your body generates, there's lots of different ways that they can bind, and you don't necessarily know what's going to happen. So
Yeah.
when they select for monoclonal antibodies, they usually select for things that tend to get bound by the most people, or the thing. So they
Yeah,
find
that's
things that
true.
so it doesn't necessarily like show the diversity of the immune response.
Yeah. Yeah, I mean like when researchers are showing the inability of a monoclonal to suppress a monoclonal antibody to suppress the virus or neutralize the spike protein or whatever assay that they're using, right? Yeah, that doesn't mean that is not that's also that's more informative. It's just giving us a landscape maybe, right, of the vulnerabilities, right? Like the places, the flexibilities of the spike protein. This is
Mm,
like when we saw the epitope mapping, right, with the ACE2 decoy, right?
mm,
Like it's showing us like a landscape across the protein, right,
yeah.
of what things could be changing to make things more permissible. And of course, like it's not good, right? Like
Mm,
it's not a good thing that they can escape these, but it's also not it's not like the nail in the coffin
mm,
for vaccines, right? Like vaccines still have value, right, in light of this evidence. But it is informing us, right, that there are places where evolution could drive the virus, right, potentially if we're not careful. So I mean, I think I've seen it several in several media venues now. So right, it should be well, people should be well aware. Even if you were vaccinated, you should still be wearing your mask and doing your social distancing,
yeah.
because yeah, it's just the virus is sort of a mutable thing. I mean, of course, it doesn't mutate as fast as other things, but given a high rate of infection, right, that's just more events for stuff to happen.
Yeah. Uh, so now
Cool.
got on a similar topic, we've got like mRNA 1.273 vaccine escapes neutralizing antibodies against spike mutants from global SARS-CoV-2 variants. So this comes from Moderna. So this and they I think maybe keep in that mind when you're reading this. They look at a number of global variants, including the B.1.7, B.1.351, but also things like the mink virus we looked at a couple of weeks ago, and a few other
Oh, fun.
variants.
Yeah.
Um, and in this one, they immunized rhesus macaques and then tested how well their serum neutralized these three different strains. And then they compared that to sera that were collected from humans after a booster shot. And they found that,
Interesting.
yeah, they could find basically that all of the variants seem to behave the same except for the B.1.351, which was slightly more resistant.
Hmm.
So yeah, it's kind of big of Moderna to say, yeah, our vaccine works except this one maybe, so maybe we need to do something about this. It still
Yeah.
works. It's just you probably
I
would
heard that there was news that the like Moderna was saying, oh, but we may reformulate, right? Like because of this whole like mRNA platform, they could just
yeah.
stick in that variant spike potentially.
Yeah, they totally could. I mean, the question is whether they want to do that,
Yeah.
because if they do that, then they want to make sure that it's still like neutralizes original recipe SARS-CoV-2.
Oh,
So
absolutely. Yeah.
that's the thing that they need to figure out, whether they can create the version of Spike that will work for both, or whether they'll
And
need to
I
like,
mean, I think the other thing is like, what is the proper like this vaccine platform? It's very new, right? Like
mm,
in terms of and it's being deployed at such a wide scale, right? Like this would have never happened, right, in a regular circumstance. So it also actually brings up new regulatory questions, like how should such like what is the standard of evidence that we want to see from a type of vaccine like this, right? That just varies this mRNA behind it, and the protein is like a little bit different. Yeah.
yeah, I feel like there is some precedent there with the influenza vaccines, which are changed on a yearly basis. So I feel like,
Right. Yeah, that's a very good point. And those are, yeah, they're just a different platform, right? But they are something the same that like there's something about the manufacturing process, I guess, of that
mm,
vaccine that gives it the similarities from from year to year, right? That reduces the amount of regulatory burden that gets put on the new ones that come out.
yeah, and still on
Cool.
the idea of viral escape mutants, we've got viral epitope profiling of COVID-19 patients reveals cross-reactivity and correlates of severity. So yeah, previous studies provide evidence that some new strains are resistant, but this can potentially threaten the efficacy of antibody threat therapies. So this
Yeah,
goes
this is,
in,
I guess, more from the research side of it now, right?
yeah,
Just varying up everything.
yeah.
Um,
They basically do a deep mutational scanning where they map all the mutations that could potent. Oh wait a minute, hold on. I think we've already
no, I,
covered this one, but
uh,
yeah, they basically like do complete maps to cover like each single amino acid mutation that can potentially fully escape the Reagan Cov 2 cocktail. Um,
mhm.
So yeah, the maps like kind of identify mutations that are selected for, and then they look for mutations in they're circulating in current SARS-CoV-2 strains that could potentially threaten these antibody treatments. Um,
Mm. And they also have like funny controls here. They have like a pre-COVID-19 era and post
yeah,
COVID. They're also looking not just at, is it that they're looking at all coronaviruses maybe? And they want to say? No, I don't think so.
uh,
Mm.
yeah, they do a lot. So they do like common cold coronaviruses as well. Uh, so yeah,
I guess
they do a fair
they're
bit of.
negligible. Yeah.
Uh, any
Yeah, it's interesting. Like, I feel like we've seen a lot of, you're right, we've seen something very similar to this before, right?
yeah.
Going across all the different. Maybe what sets this one apart is that it's not just Spike. They end up focusing on Spike in this paper,
Mm.
but they did end up scanning through everything
Yeah,
on the.
that was quite an important idea, I think, because we do get very spike focused because that's very important. But they look into nuclear protein and other things.
Yeah.
Yeah.
Hmm. Okay.
Next one is genomic monitoring of SARS-CoV-2 uncovers an NSP1 deletion variant that modulates type 1 interferon response.
Yeah, I thought this was a very odd uh, paper to come up like that. NSP1. We had actually a structural paper on NSP1 for a while, but now I guess they found some other mutations in it, and this may be a story of things getting viruses becoming more acquainted to their hosts.
Yeah.
Finding that sweet spot where they can still replicate inside of a host, but not cause disease. I mean, they don't really talk about disease here, but the mechanism that they're describing is that pathway to being able to infect people, but not tickle the immune system too much. Yeah.
I mean, I really like this because of the way they kind of collected the data. Because what they did was, I think they looked in January and February in hospitals in China, and they analyzed the genomes collected at that time and correlated that to those symptoms of the patients. And then they picked out the ones which like had unusual symptoms or symptoms that were slightly different, and looking to see
Yeah.
whether like parts of the genome correlated to those symptoms. Which again, it's kind of there's a lot of things that can go wrong with that kind of analysis. There's a lot of
Hmm.
confounders, a lot of details that can mess up, and they take that, and from that they picked out some mutations,
Yeah.
and then they took it into the lab. So this is what is known in the lingo as bench to the bedside, or bedside to the bench, I think is where.
Yeah, it's like the opposite. Yeah. But yeah, I think that's really that is an interesting observation in like the way that they use their data. I like it. It's like it's a new hypothesis, right? Like the method, even if this ends up not being like a very important thing, I think
Hmm.
the methodology that they use gave us like something very relevant, right? A very relevant hypothesis to pursue, right? Are there changes that will be happening to the virus? Are there changes that happen to the virus that yeah, modulate the disease in some way. And what does that say for its trajectory in evolution in our bodies?
Yeah, I mean, I think I'm not sure I'm comfortable with it because it does feel very much like a genomic fishing expedition where like there are like kind of when you see those psychological studies where they try to find the gene for you feeling slightly hungry after six. I mean, it's
Yeah,
like
yeah,
I do feel like there is some element of that, but I mean they do confirm it by taking it to lab experiments. So
right,
it's
right.
but I
But I
find
mean, they don't take it, but they don't take away, they don't take it all the way back, right? Like
yeah.
they do some bench stuff, but it's like once it goes into the bench, it's really hard to then like go build the complexity back up and say this is what is causing it in humans.
Yeah, that's right. Because there is so much complexity there, and so they pick up one thing out of a very complex system, and so you're not necessarily going to see the whole complexity of the system encompassed by just one thing, but that can still be quite an important thing that you're looking at. Um,
Yeah. Okay. I think
yeah,
that's for that paper.
yeah. Next paper is viral epitope profiling of COVID-19 patients. Let me put this
I
in.
don't know. We just
Did we?
Oh, well, no, they're just slightly different titles.
Yeah. It's, so I
Right,
didn't.
the one that yeah, we already looked at the, I see. It's yeah, because this star one that we had already looked at, and it's this one is new. But I'd been looking at this while I was looking at the one before. My
Oh,
mistake.
right. Okay, right. That's okay. But so the one before I feel like wasn't as good as
The
this
one
one.
before
So
we had seen before, and that was they made those mutations just in yeast.
yeah,
Right. But this one is different because um they're actually looking in
yeah,
to find the things that are mutating, trying to figure out what is changing.
yeah. And this is and
Not what's mutating, but like from all the mutations that come out, right? Like what correlates to different severity of disease. It's kind of a very machine learning things, and this is where I said, sorry, that's why, sorry, I misspoke for the last one we were covering the star because I was looking at this, I was looking at this paper, right?
yeah, they are very very similar. I'm just like double checking like mad because in case I've got the like wrong paper, but
Yeah,
because they
the Schrock paper is the one where they can find things other than spike in this type of approach.
yes, yes. So
But the
yeah,
star
this is
paper they could only find things because in the star paper it's very it was very bench science, right? They have the ability to do the deep mutational scans. Here it's like, oh, all that is natural mutation that happens in the population. Let's correlate that to some patient data that we have.
right. Yeah, whereas this paper is so I
And
think I might do yeah.
I think
Sorry.
it's actually about their machine learning model mostly. It's
Yeah.
actually, this is like some, oh, look at this new way of reducing this very complex data set, right? If you imagine like all the different mutations across every infection, how do you find the ones that are relevant, right, to some sort of patient data that comes out from that? I guess they're looking at antibodies being made against different things.
Yeah, so essentially they're trying to find that commonality in the antibody responses. So they do like some phage profiling and they find that there are like various correlates that there are various areas of the virus genome that do cross-react. So it's kind of interesting because it does go beyond spike. So they look in look at spike and they also look at kind of other proteins that show up.
Yeah.
And
Yeah.
yeah, I'm not clear fully on the machine learning, but but they've got a lot of data that they're dealing with. So and it kind of churns through that to create an idea of what's happening.
Yeah, absolutely. And sorry again, like the things I was saying for that star paper, I was saying while I was scrolling through the figures for this paper.
I'll be honest. When I just realized that the paper I showed on the screen was actually this paper instead of the star
Oh, okay.
paper.
Oh, so actually it
So
was weird. We are kind of on the same
kind of
wavelength.
on the same page, well, same
Okay.
wavelength.
Okay. Okay. As long as people who follow along are not super confused.
Yeah, I'm so sorry, audience members who might be confused.
Okay, let's
But
see what else we got.
we got CD8. So CD8 T cell epitope variation suggests potential
Hmm.
antigen presentation deficiency for spike protein of SARS-CoV-2. So we we've been focusing a lot on antibodies, but there is a whole other aspect, the cell-based immune system. So T
Yeah.
cells are very very important for viral responses.
Yeah, and they
Uh,
have their own repertoire of things that they recognize. So antibodies are the recognition sites that are on B cells, right? The B cell receptor is essentially the antibody, but T cells, they have the same type of variable. Well, they have a similar type of variable receptor, right? That gets selected for. And yeah, here's what it is in T cells.
yeah, so T cells rely on the binding not just with the antigen but also with like this thing called the human leukocyte antigen or major histocompatibility. It's called many things. But essentially, the function of this.
The way I always explain it to people is I think a lot of people have heard about this study where like if people sniff different clothings, they're like more attracted to people with different HLA types.
Yeah,
And like that's what it is, right? Like and one of the hypotheses that came from that is like, oh, it could be for immunological like robustness, right? To mix all the HLA types. But basically, yeah, there's a protein that's hard coded in us, and it presents the antigens to T cells.
yeah. So in your cell, you've got proteins, and occasionally they need to be recycled and broken down. And when they're broken down, some of that protein gets expressed on the surface. And this little protein takes a peptide of a certain length, and they present it on the surface. And the T cell can receptor scans it essentially to see whether there's anything unusual. And so it's very good for spotting viruses that are replicating foreign virus particles in the body, and it's also good for spotting
Because it
cancers.
shuts down, yeah, it shuts down that infection at the site, right, at the source of the infected cell, right? Because we know that the viruses replicate inside the cell, so it's like just like, oh, this cell is infected, we should just auto self-destruct it.
So yeah, just taking that into account. Think about if you're a virus, how would you try to get around this? Well, the obvious way is to try and stop your proteins from being expressed on this. So having a spike epitopes that are less able to be bound by this HLA.
Yeah,
So
yeah, it's like another arena of host pathogen evolution arms race, right? Where it's like, oh, we don't want to be picked up by these MHC proteins, so let's have different sequences and yeah, but still have to remain functional, right?
yeah, so what they find is that some mutants seem to evolve to not bind HLA. So in convalescent patients, that they didn't have a specific TCR to recognize these, but uh, the idea I think was that yeah, that there seems to be some evolution away in SARS-CoV-2 from this. But even then, it's interesting because then when so like while they didn't have T cell expressors that kind of recognized some of these new peptides, they did
Mhm.
the T cells were still able to mount a response. So
Right.
it's like kind of so so yeah, and it's an interesting look at kind of what's going on
Very,
with this.
very, yeah, like there's a lot of detail, right? In terms of like anything epitope mapping is going to be very detail oriented because it's about very
Mm,
specific regions. But I guess the information that could come from it is um, maybe learning about, I guess that this is actually learning about that other compartment, right? Like what's important in that compartment in infection. Maybe, right? Because everyone's been using these like spike protein neutralization assays, right?
mm.
As the de facto our thing works, our thing doesn't work as like a first pass. Maybe this type of study could influence like another type of first pass experiment, right? Where it's like,
Yeah.
let's see if it induces like a certain T cell, if it's recognized by these T cells that we've grown up, right? That like we know these are the important epitopes to be recognized for productive infection. Let's test that in our vaccine responses.
Right. And I think another thing so I think I want to talk about the next paper called Comprehensive Analysis of T cell immunodominance and immunoprevalence of SARS-CoV-2 epitopes in
Oh yeah.
COVID-19 cases. So that kind of delves into it a bit more detail because it looks at, say, what are the T cells responding to? What are the common kind of, where are the weak points of SARS-CoV-2 when T cells are recognizing them?
Yeah.
So,
Yeah, so
so
like, the last paper was like the full, like just like a broad everything, right?
yeah,
And it's like let's find some significance maybe out of that information.
yeah. So this looks at the whole genome of SARS-CoV-2 and tries to figure out which dominant genes are there because they're not necessarily, for this, it's not necessarily going to be a spike because it could be any gene that the T cells can react to.
Hmm.
Looking at what the common like epitopes are there, and they're also comparing that to the antibody responses, seeing like how this relates to the. So if like an antibody reacts to an epitope, does that mean that T cells are more likely to react to the same thing because of the way
Yep.
that the immune system talks to itself?
Okay. Up next, SARS-CoV-2 infects and replicates in the cells of the human endocrine and exocrine pancreas.
Yeah.
Yeah, I guess like we were talking about like the NLP1 that's like expanded, right, repertoire of
Mm.
things it could affect. I guess they came to this hypothesis because there are some metabolic dysfunctions in severe COVID-2 patients,
Yeah.
in COVID patients. So they went in and they did some staining of a bunch of infected pancreas, and finding that, oh yeah, they have ACE2, they have TMPRSS, what
Well, I
TMPR,
guess.
yeah.
Yep.
So we know we can get entry, and looks like there is some pathology because of it. It seems like is what they've looked at here.
Yeah, that's interesting. Another insight into like potential side effects of getting COVID and long term effects as well.
Oh yeah.
Um, So yeah, that's an interesting one. Next one is human erythroid progenitors are directly infected by SARS-CoV-2. Implications for emergent erythropoiesis in severe COVID-19 patients. So
Yeah, very
um,
similar again, just another tissue type, right?
yeah, so in
These
this
are the
case, Yeah.
Oh, go ahead. Oh,
Oh yeah,
tissues
they
that make
look.
red blood cells.
Yeah. Uh, yeah, so I think what they found was this thing called stress erythropoiesis, where if your blood cell levels are getting low, your body starts pumping out more and more like blood cells, and some of them turn out to be nucleated. So I think they found like more circulating nucleated red blood cells, indicating that there could be something going on with the stem cells. So they take a look at the stem cells and find that actually SARS-CoV-2 can infect them, and that's
I think this is
interesting.
this is like the huge challenge, and we spoke about this in I don't remember which one we spoke about it now, but like definitely one of our deep dives we spoke about this idea that like ACE2 is in all these different spots of the body, but the symptoms that come out don't necessarily map onto that. Oh, it must have been the NLP. It was the receptor one, right?
Yeah.
But this is like even knowing that where the receptors are, then you have to study every tissue type, right, in combination with SARS-CoV-2 and come up to some idea. So, I mean, this is where, like, yeah, seeing symptoms really is helping to drive these hypotheses, right, and this exploration to drive down to like a different mechanisms that could be happening during infection.
Yeah, I mean the whole pathogenesis can be really complicated because you are dealing with so many different humans have so many different tissues, and how SARS-CoV-2 or even any microbe reacts to it can be very very different, and it can produce. Always a diversity of symptoms in any disease, so
Yeah.
I think this can highlight that kind of picture. Uh,
Okay. SARS-CoV-2 ORF9 inhibits RIG-I MAV's antiviral signaling by interrupting K63 linked ubiquitination of NEMO. So very
yeah,
logical, right? These are specific immune pathways,
yep,
RIG-I MAVs, Ki63 linked ubiquitination of NEMO. Those are innate immune pathways that are well known. And here is a SARS-CoV-2 protein that disrupts one of these things.
yeah. I mean, this is quite an interesting one because like RIG is very important because it recognizes double-stranded RNA. So
Hmm.
that's any virus wants to knock that one down. That's like the big thing that so yeah, and that rig starts like kind of this conga line of messaging, and
Yeah.
apparently what Orph9 does is knock out one of those like kind of links in the chain, so to kind of stop that signaling cascade.
Hmm.
So it'd be quite, and they do seem to go into a lot of detail of like how this happens. So that seems like be quite an interesting one.
Yep.
Uh,
And we got loss of furin cleavage site attenuates SARS-CoV-2 pathogenesis.
yeah, I feel like we've talked about this a fair bit.
That's right. Yeah, we've already. I was
Yeah,
gonna say this is something that we've seen before. Both because when you passage the cells, right, like they lose that furin cleavage site. Uh,
yep.
so like it's important in the human context, but not in others. And yeah, I think yeah. So if you don't have it, the disease just doesn't go off as well.
Yeah,
Um,
the interesting they do like deliberately infect Vero cells with like a mutant they created. So they actually like went the effort of creating their own mutant rather than pass out, and they found that like it is doing something different in Vero cells,
mhm.
but it doesn't really they don't really delve deeper into that because uh,
Yeah. And I guess maybe the one reason, I mean, I did it because it's like, oh, it's kind of like published in this high pathogen
hmm,
we've talked about before. The other reason I thought it was just interesting to highlight is to reinforce, I think it's the NLP1 to NLP1 callback, right? That like
hmm,
that furin cleavage prepares
yeah,
the spike in a specific way, right? That may lead to pathogenesis, right? It opens up another floppy end, right? That could be binding
hmm,
something, such as NLP1.
yeah, the
So just to
CN
remind about that and point that this will be seen in other research, right? Like this
yeah,
is the that incarnation of it seen here.
yeah. Uh, Next one. Oh, sustained cellular immune dysregulation in individuals recovering from SARS-CoV-2 infection.
Yeah, so this is like some long term. This is back to the long COVID story.
Mm.
Uh, so,
Yeah.
I guess with the pancreas, we're talking about a different cell type, but what about just like long-term effects on the immune system? Because like the immune system has a lot of things that keep it in balance. And so here, I guess they're looking at different T cell activation markers, exhaustion markers, and showing that they remain elevated after infection in a strange way.
Now that is interesting because we did touch on this when we were talking about the B cell So about like the B cell development and how SARS-CoV-2 can mess with that.
Yeah, the germinal
So
centers get messed up. They like, yeah, like during infection, basically they're not going down the regular path, right? They become something different, something that's able to make antibodies towards all sorts of things potentially. Yeah, and this would be like another piece of that because T cells also play a role in that as well, right?
yeah,
In germinal centers.
yeah, I think yeah, that's quite an interesting one because I mean it also emphasizes that this isn't just about getting the immune response; it's getting the right immune response. And SARS-CoV-2 survives when it causes your body to have the wrong immune response, which is
Exactly, exactly. And actually, I mean, just and now I'm just kind of spitballing, but like it's also a fun thing to think about, like that that kind of crazy immune response, right, is giving the opportunity for SARS-CoV-2 to infect more people, right, to spread
mm,
out and get more of its replication happening. And maybe in that mix, it's trying to find, well, it's not trying to find, but like nature will be pushing it towards mutations that let it like find that sweet spot. Right, so like it's in there, this like initial ability to suppress the immune system and cause some crazy pathogenesis is giving it the ability to then also continue down the evolutionary path and maybe just become some long-term infection that people get.
yeah,
Okay, that's just a crazy crazy thought from reading all these papers.
yeah,
Okay, the next paper that I want to present for potential deep dive is D-dimer-driven anticoagulation reduces mortality in intubated COVID-19 patients, a cohort study with propensity-matched analysis. And really, it's just a follow-up to last week's deep dive.
yeah.
Saying that, oh, well, we talked about some mechanism last week, and here's an example of how those mechanisms drive therapeutic interventions, right? They had proposed in that complement thing that, um, yeah, this D-dimer, the accumulation of D-dimer and improper coagulation is a cause of certain negative effects of COVID-2, and here they're directly acting on it.
Yeah, so they're basically seeing whether this is a very clinical study, although it's not a clinical trial, it's not randomized, it's a retrospective chart review, which is kind of one of those things that is quite good to do if you have access to all this data and you want to find something else about it and you know that some people get treatment and some people don't because of certain bad things. So from this they can there so yeah there are always like a bunch of asterisks for this kind of research, but it's also quite kind of interesting because this is very treatment oriented and we tend to be very like kind of very sciencey rather than
Yeah,
looking at treatment outcomes.
but I did want to like I just want to show maybe that interplay, right? By
Yeah.
paper, it's like, oh wow, like in early of, I don't know, October was it that
Hmm.
paper came out that we covered last week? There was stuff about the mechanism behind it, and that gets people thinking, right?
Hmm.
And people thinking means, oh, look, and all these doctors have access to this data. Let's see if it has any effect, right? If we looked at just the way people were being treated normally, can we see some patterns in that? And it's very much the science of medicine, right? Like medicine
Yeah.
is its own set of like rules and like thoughts about like how best to take a patient through a flowchart. And these
Hmm.
types of studies attempt to, modify that flowchart slightly. And of course, there has to be more follow-up studies on stuff like
Oh,
this in order to really permanently change that flowchart. But yeah, it all works together. The
but yeah,
hypothesis
it's
generated by mechanism seeps into medical work.
yeah, and I think it's something to keep an eye on in future. I think because this isn't just something that will just be solely for SARS-CoV-2. Lots of other like diseases also end up causing a hypercoagulation cascade. So
Mhm,
it's something
mhm.
to think about. So I mean, I'm going to keep an eye out for trials on this because I guess for SARS-CoV-2 we have that much more data now on one
Yeah,
disease, so we can pick out these sorts of effects that we can
absolutely.
make take out to other rarer diseases.
Mhm. Okay, and the last one on the list I think we have is active surveillance of asymptomatic, presymptomatic, and oligosymptomatic, which I'm not really sure what meant, SARS-CoV-2 infected individuals in communities inhabiting closed or semi-closed institutions.
Well,
And this is really about sampling protocol or, yeah,
yeah.
screening protocols. Yeah. Like, how do you detect outbreaks very early? Long-term care centers, prisons, places where there's a lot of mixing. People want to detect early, but you
Mm.
also don't want to have to test every individual person. Schools fall into this category too, right? You don't want to have to test every individual person every day to figure out when the outbreak occurs. So they compare two different pooling methods in this to show which one might work better.
Yeah, so it's interesting because like you have one option of like testing every single person, but tests aren't always that sensitive to very small amounts. So the idea is that they get instead of testing each individual person in the room, you take those tests, put it all in one bucket, and test the bucket.
Yeah.
And
And then
if
if
that
the bucket
bucket,
shows something, then you have to retest all the people in the bucket, or there's like a cross pool or something like that. Everyone's randomized. It's actually really similar to epitope mapping. I don't know if anyone
yeah,
remembers this, but it's a very similar approach to how people do epitope mapping.
yeah, I think the interesting thing is like is it because the effect efficacy of this they do talk about this in a little bit in the paper where like it depends on how much like pre-test. So if there's a lot of positive people already in the population, this doesn't actually do that well. But if it's rare, then what you're doing is trying to like isolate people and try to clear people as well. So you can
Yeah.
so it might not be able to find an individual person, but it can clear a room of people. You can say, okay, these people don't have the COVID, and that's
Yeah.
fine.
Yeah. And
And
it's really
so
important at the beginning of an outbreak, right? In these large facilities, right? You'd be like, oh, floor one, that's the problem floor. Let's institute some important
yeah,
regulations to prevent it from spreading to the other floors, something like that.
and if you're short of tests, it also is very useful in that regard. If you don't have enough like mRNA reagents or things like that, then actually this can save you on resources if you're being stretched.
Yeah, absolutely.
Um,
And I guess maybe I just want to say before we try to choose a paper is the other reason it's nice to even close with this paper is because like you can't stop plugging it enough that like even though the vaccine's out there, right, like we still in many places have rising cases, right, and like in those circumstances like it's important to be able to control outbreaks, right, because we don't want to be basically vaccinating as fast as we're getting infections. Like even though like yeah we're saving people from severe disease, there's all these long-term effects of COVID. We just don't want so many people having this virus, and there's that potential for mutation to occur, right, in all these scenarios. So testing is still really important, and emerging information about testing methodologies is also still very, very important.
yeah, for sure. And I guess now it comes to the point of the show where we have to choose. We have to pick which one of these great papers we wanted to take further.
Yeah, absolutely. I think from this round of things, I was most interested in either of the either the pancreas or the erythrocyte one, or maybe the immunodominance of T cell antigens.
Yeah, I mean, I was quite interested in the immunodominance of T cell antigens one. That seems like that'd be quite an interesting one to dive into.
Yeah,
Um,
I like it too because setting up from the idea of everyone's been using neutralizing antibodies. We've seen so many neutralizing antibody tests now,
yeah,
right?
yeah,
It would be interesting to think that there's some sort of alternative to that, right? That could tell us about whether something is effective, whether a vaccine is effective or not.
yeah. So for me, it was either that one or the sustained immune dysregulation because we haven't really covered long COVID very much, but I do feel like those. But yeah, again, I was on a cellular immunity kick as well, so
um, and then I don't know if we're interested in any of the other older things. We still have uh, secondary structure across the SARS-CoV-2 genome. We still have the real-time conformational threat of the spike protein as it sits on the surface of viral particles. Um, MASP. I've been kind of moving some of these out as like
yeah,
we've been seeing other things. Oh, there's still those. I'm actually
yeah.
feeling immune. We didn't do immunity last time. Well, we did complement. I don't want to I'm trying to think. Do I want to do rig? Oh no, no, you said the long-term COVID. That was not the rig I one. That
No,
was
that wasn't the rig. That was the let me pull up the title quickly. It was the sustained cellular immune dysregulation in individuals recovering from SARS-CoV-2 infection.
oh,
So
it's right here. Okay.
yeah, so this is essentially they use tons and tons of flow cytometry to look at cells, looking at the stuff that's on the cells, and then trying to figure out what's been happening, what's going on, what is going on in the mind of these cells. how they reacting afterwards, after the infection.
Mm-hmm. Yeah, that's interesting. I'm like back I maybe I'm back in the I want to talk about mutations again, or like the like what mutations may impact
Hmm.
epitopes is something that gets recognized, right? It's talking about the
Okay.
structure of that. I'm feeling the immunodominance paper.
Okay. Yeah, I think we can go with the immunodominance paper because I feel like mutations are quite in right now, and it'd be quite. And what I'm hoping for is like some kind of idea of like whether the the mutations that we're seeing in the B cells right now in like the antibody responses are going to be reflected in in the T cells. Are they going to cause the same kind of problems? And I'm hoping that this will
Yeah.
get me more understanding of that.
Yeah,
And
maybe they do seem to map out some of the epitopes across the whole genome, right? Because it could be
yeah,
anywhere. Right? That's the other thing. Maybe it's nice to talk about that distinction too, because we've been stuck inside this, always talking about spike protein. What are the antibodies going to do for us, right? How are
yeah,
we recognizing and stopping the fusion? But this event is like the virus already got in. It's fused. It's inside of a cell, but you want to prevent that cell from making more virus. There's actually a lot of different places that that could happen over
yeah.
the
I
gene.
mean, I think that it's because you're stopping the actual virus factories from where it's coming from. That the T cell response is really because almost when the virus is already in your bloodstream, the damage has basically been done. But if you got getting at the cell level, then you're like, okay, if I neutralize one antibody, I've neutralized one antibody. If I kill one cell that's producing millions of antibodies, then I've done more of an
Millions
effect.
of virus. Yeah, absolutely.
So that's why I'm quite interested in looking into T cells, seeing what's happening. I am aware that like also T cells is, I mean, the cellular immune response is also where I'm quite weak because there's so many different like surface receptors to keep in mind when we're looking at these sorts of studies. So
Yeah.
it's going to be a bit of a challenge as well.
Hmm. Yeah. If that's okay with you, then that's what we'll cover. Comprehensive analysis. So for next week, we'll be covering in our deep dive comprehensive analysis of T cell immunodominance in oh gosh, I have a dot 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 now.
Oh,
And
in
immunoprevalence of SARS-CoV-2 epitopes in COVID-19 cases.
yes. Okay, yeah, that's fantastic. Thank you for joining me for this. It's been great talking to you about these sorts of crazy papers and new science that's coming out.
Yep, absolutely. Just to remind everyone that we both believe that peer review is a process that we can all participate in, and we're doing our best to give you the the best information we can. But we may have made a mistake, so if we have, just let us know in the comments.
Yeah, and you can also reach out to us over Twitter with the hashtag microtwjc. Uh, and we're both very enthusiastic about microbiology, and tune in more week for more microbiology content where we'll be talking about not just microbiology but immunology. So
Yep. Great to chat with you, Fuzz.
yeah, great to chat with you too, Danny. See you next week. Bye.